I will be talking how we do in my lab in Düsseldorf, Germany, to model Leigh syndrome using induced pluripotent stem cells and derived neuronal cells and organoids, which is here you can see an image on this side. So as I'm sure this audience might be aware, Leigh syndrome is a frequent and severe manifestation in children of mitochondrial disease, and can give neurological symptoms like you see here, MRI depicting sort of a typical bilateral calcification in the basal ganglia. And this causes a disease of movement and also a mental regression. And there's no treatments available. Genetically, we know the cause. It can be caused by mutation in several genes that can be in the nuclear genome or mitochondrial genome. And all these genes are involved in the production of energy by the mitochondrial respiratory chain oxidative phosphorylation system. And today I will be talking about two of the genes that are known to be affected, which is one Surfl, which is a complex an assembly factor of complex 4, and ATP6, which is instead a component of the ATP synthase of complex 5. So, like we all know, we don't have therapies for Leigh syndrome or mitochondrial disease in general. And we know that this is a challenging aspect of first to pursue. I just want to highlight one particular, some of the challenges when we think about the drug discovery path. We have to start for identification of target and then do preclinical phase in different models before going to study in humans. And these can be challenges for mitochondrial disease where some of the tools might be more difficult to have for mitochondrial mutations. So it's less easy to have overexpression systems or knockdown or animal models that are maybe available for nuclear genes, or other animal models or cellular model might not be recapitulating a feature of the disease. So the path that we're trying to follow is the one to use induced pluripotent stem cells, which can be generated from patients and from controls. And these can be then differentiated into the desired cell type, like in this case, cellular of the nervous system for Leigh syndrome, I mean. And there's two challenges. So we need to see whether these cells can exhibit a phenotype that can we use to understand the disease, but also as a way to perform compound screening. And then the second step, it should be done in a way that there should be cells that can be used in a reliable way for compound screening. But if this path could be used, there's a potential that maybe can lead to identification of therapies. That's basically our hope. So we know we've been working with iPS cells in several years. We know what happens when we make iPS cells from fibroblasts. We know there are changes in the gene expression profile. There are changes in metabolism, as in iPS cells are less relying on mitochondrial metabolism, so they tend to, after pyruvate, to tend to produce lactate. But we also know when we start from cells with mitochondrial mutations, so here there will be a case with the mutant mitochondria, we know that we could have iPS cells with more or less mitochondria, and this we described 10 years ago, but now recently also work from Valeria Tiranti and Patrick Schinery recently also showed the importance of this. So also in the context of control cells. So basically what we do in the lab, so we have from patient that we have in collaboration with clinicians, we generate iPS cells, and then if the mitochondrial mutation, like I said, there's challenges in repairing, but if there's a nuclear mutation, we can use genome editing to repair the mutation, and then we generate a model system like neurons, NPCs, or brain organoids, and then we try to identify differences of phenotypes, and then to potentially find the therapy. So what are brain organoids? So basically, when we start from iPS cells, basically you start from aggregating the cells, and then you let them cells spontaneously differentiate. And there's basically two strategies. One, to basically don't do much, just keep these organoids, growing organoids in a bioreactor, so to let them grow over time. It's in a more physiological approach. Or you can add on top some small molecules or growth factors to push the organoids in a defined direction, for example, dorsal forebrain or midbrain, so to have more defined identity. And then you can even fuse this in so-called assembloid. So this is the strategy that we're pursuing, because we want to have organoids from several patients and control. So and this strategy is more, there's more consistent, as the organoids are maybe a bit smaller but more equal. While in the first option, there is increasing complexity and diversity, and which can be more difficult for our approach, but maybe good for in other contexts, like for example in understanding really developmental aspects. So these organoids, they're made in a dish. So here we see, for example, if we have 60 days organoids, they might correspond to the second trimester on human prenatal development. And this is important to consider of the limitation of the system. So it's not good, let's say, to study age-related aspects. And also there's no vasculature and there's no microglia. So aspect related to immunity are also not possible to investigate. So what we do, we do that, and we put the cells in an orbital shaker, so that plates in an orbital shaker, so we don't use the bioreactors. In this way, we can scale up. And so, for example, a plate here, you see these small roundish aggregates, the brain organoids. And then the important thing is they can be functional. So we can see here these yellow triangles. These are supposedly spines that indicate synaptic occurrence, and we can use calcium imaging to see that the cells respond, for example, to glutamate addition. So we use this system to investigate Surfl mutation, like I mentioned before. Surfl is an assembly factor of complex 4. So here, Surfl help assembly of complex 4 in the mitochondrial respiratory chain. It's a nuclear mutation, so there are animal models available. However, some of the features of these animal models might not completely recapitulate the features observed in the patient. So we think there's a need to have a human model system to investigate the disease mechanism. So since it's a nuclear mutation, so Pavel Apostel in the lab could use CRISPR-Cas, and we could have iPS cells from control where we introduce the mutation, and iPS cells from patient where we corrected the mutation. So we have then these different patient and control mutation in different backgrounds. And we see that the neurons fire less action potential, but then they also in the organoids, there's less differentiation of neurons. So then we try to understand why. And so we did the single cell transcriptomics of both neurons and organoids. And we saw that in the Surfl neurons or organoids, there's a difference, a distribution of cells in the cell cycle. So there's less G1 and more G2. So the cell seems to proliferate more. And we look at the, we combine transcriptome metabolomics and proteomics to identify signature, and we saw signature related to cell cycle and metabolism, also to synaptic dysfunction, but also to morphogenesis, so to process, to generate neurons. So then we went back to understand maybe there's something in the development of neurons that it's occurring. So the organoids from the controls are bigger than the one from patient, which we found interesting, as in some cases, people with Surfl mutation also can display microcephaly, so smaller brain. And then we look in more detail at the organoids, specifically at the part of the progenitor cells that generate the neurons. And you see in the control, there's a nice organization and distribution of these LHX2, which are progenitor, or SOX2 progenitor. They make these neurogenic zones and phosphovimentin at the apical layer of the progenitor of the neurogenic zones. But this is basically disrupted in the Surfl organoids. So the organization is disrupted. So basically are smaller because they are not able to make the proper development development. So then we went back to 2D to understand if we can pinpoint a specific defect. And we saw that in the neuroprogenitor cells from control, they start to make branches because they start to differentiate, but in the Surfl NPCs not. So we could quantify this defect in neural length and branching point. And then we use this as a way to understand if we can find like a phenotype, like I mentioned before, to find therapies. And we observed that by using, for example, bezafibrate, which was increasing the ATP production of NPCs, we could observe an amelioration of this branching possibility. So to summarize this part, so we think that during the process of making neurons, NPCs should switch more to OXPHOS, so should become more dependent on mitochondrial metabolism. But when this is not happening, this could be a target of intervention. And for example, PGC1 alpha activation by bezafibrate could help improving OXPHOS capacity and improve the NPCs to shift to mitochondrial metabolism, and so to differentiate into neurons. So that means that we can use brain organoids to dissect mechanism of the disease, and for example, also to highlight components. There are neurodevelopmental components of the disease that we might be able to target. But I just want to take the last few minutes to say the importance of that, and also in the context of other Leigh syndrome mutation. So because we see it's much more complex. This is a neurons, astrocyte, and blood vessels. So there's a big metabolic exchange, and the presence of mitochondria there in neurons, and the presence of mitochondria there can allow that. But maybe not only neurons, but also progenitor cells might be affected, like I mentioned before. And what we found before is that, in fact, this is iPS cells. These are the mitochondria in iPS cells. And these are mitochondrial neurons, which are more mature. And we found that already neuroprogenitor cells have mitochondria with a dense cristae, which is cristae are the site of OXPHOS, so suggesting they are depending on OXPHOS. And in fact, when we measure glycolysis, we see that it's higher in pluripotent stem cells. So when we measure lactate production by ECAR, we see that it's higher in pluripotent stem cell and lower in neurons and NPCs, suggesting that NPCs instead, like from pyruvate, also to use the TCA cycle to generate energy. Sorry, I switched this. So then we decided then to use this system for ATP6 mutations. So we found that NPCs from control are able to respond to depolarization of mitochondrial potential to release calcium. So here see, but in the patient, NPCs with ATP6 mutation was not occurring. So ATP6 is another common gene affected in Leigh syndrome. It's a mitochondrial gene. It's in a component of ATP synthase complex 5. So this is just a quantification that the control are able to have this calcium release, but the patient less. We identify another phenotype related to mitochondrial membrane potential. It was higher in the patient compared to controls. And so, and basically to summarize this part about ATP6, we think that this defect in the MMP would cause a defect in calcium, and that could represent a screening target. But this time we don't only tested a few compounds, but we sort of try to develop more of a high throughput screening. So Annika Zink in her lab, so we developed this high throughput screening based on high content microscopy. So you can measure MMP, and then you can also test that with the defined drugs, depolarizing and hyperpolarizing, we can have a Z factor, which is a way to say the window of opportunity to find compounds. And this is basically the approach they were using, and then now we have a consortium funded by HAPRD for rare diseases, where we can then use NPCs with ATP6 mutation and screen for compounds and use this MMP assay. And we have some initial results where we found one FDA-approved drug that were now being tested in compassionate treatments. So the first treatment has been spearheaded by Markus Schulke in Berlin, where there was a 16-year-old boy that had a metabolic crisis and was sent basically to die. And then there was nothing to be done. So Marcos decided to use a compassionate treatment, this compound, and was very good results. And so this was actually recently went into television in the German television as a save with a good diagnosis, you can save life. And then because of this, let's say, success, we have now 2 other compassionate treatment, one in Düsseldorf run by Felix Distelmeier, one in Munich by Thomas Kloppers. We also relatively older individuals, so 21 and 17-year-old, and this is also showing promising effect. So we hope that then we can dissect more the mechanism of this compound and also find other drugs. And so with that, I conclude saying that we think that, yeah, organoids can be important, but also we can pinpoint also some role of progenitors in the that might be important for mitochondrial disease and can be used as progenitor cells for developed compound screening. With that, I thank people in my lab, collaborators, and all of you for the attention. Thank you.